right, we'll take just one more minute. Thank you for joining us. All right, it looks like our attendance is starting to taper off, so I'm hoping that everybody who is joining us has made it into the webinar. Hi, everyone. Thank you for being here today. Good morning, good afternoon, depending on where you're joining us from. My name is Kelly Andrew. I'm relatively new to HDSA. I joined the team in March of this year as coordinator of research and mission programs. I'm very excited to be joining you all today for our Ask the Scientist Anything webinar with the editors of HDBuzz. Just a couple of housekeeping items before we get started. This webinar is being recorded. I'm sure you all got the message as you were logging in, but we are recording and you may find the recorded version of this webinar on YouTube after it's concluded, as well as all of our other research webinars from the year and previously. This is an open Q&A webinar with some HD experts today. So you may ask questions throughout the webinar at any time, and our panelists will go ahead and answer those for you all. To ask a question, go ahead and click the chat function in the toolbar at the bottom of your screen. And you can type in your question and hit send at any time. And those will only be shared with our panelists today. Just a couple of other housekeeping items. If you are interested in learning more about HD research, we have several resources for you all. One of them is HD Trial Finder. It's HDSA's clinical trials matching service. So you can log on, create a profile to match to trials, or you can just log on to view trials all over North America. And there's a call center if you have questions that is staffed by clinical trial navigators who are specifically trained in Huntington's disease trials. And it is updated regularly, so you can try rematching at any time as well to see if you're eligible for any new trials. With that, I'm going to hand it over to Eric Johnson.
Great. Kelly, thank you so much, and welcome everyone. Good morning, good afternoon. Thank you for joining us here today. As Kelly said, my name is Dr. Eric Johnson. I am the Chief Mission Officer for the Huntington's Disease Society of America, and really excited to be joining you here today. This is my first month in the role, so really happy to be able to share this webinar with you guys, especially with the great team that we've got with us. So as Kelly was saying, we have some resources that we want to make sure everyone's aware about. Of things that you can take part in or review to get updated information on what's going on in the world of HD science. First, if you want to learn about what's going on in research, please check out HDBuzz. I know these guys can talk a little bit more about it, but it's a fantastic resource for up-to-date and really user-friendly information as to what's happening currently in science. Another resource is the HDSA This Week in Huntington's Disease Research, which you can find on the HDSA website, hdsa.org/blog. Engaging in research is something that we encourage everyone to do to their level of comfort, and we hope that you do it because it's because of people who do participate in research that things are happening. So we encourage you all to check out things like Enroll HD, look at HD Trial Finder, as Kelly mentioned, and look at, if you don't already know the Centers of Excellence program, look into those as sites where you can participate in ongoing clinical trials. This is our last webinar of the year, but we want to get you aware of what's going to be coming in early 2022. So we do have a webinar on the 11th of January with Allison Bartlett, who is a specialist in disability law, specifically for people living with Huntington's disease. So if you can join us on the 11th of January, that would be great. And stay tuned for more updates as far as research and HDSA and me webinars in the new year. Another place you can find this again on the hdsa.org website. If you click on news or events, you will be able to get information on those upcoming things. And that's enough for us. We want to turn it over to the team who has joined us here today. This is an Ask the Scientist Anything webinar, and it's with a team that are the editors for HDBuzz. You may be familiar with them, but if not, we'll give you a very quick introduction and they can take it over from there. We have Dr. Jeff Carroll from Western Washington University, Dr. Ed Wild from University College London, and Dr. Rachel Harding from the University of Toronto joining us today. Guys, thank you so much. I'm going to disappear. Kelly and I are going to go off the screen and it's all yours.
Thanks, Eric. Hi, everyone. Really great to be here. Great to be back. Sad to be here virtually, but glad we can be with you anyway. And really pleased that we've been able to offer these Ask Us Anything sessions more frequently than previously. One thing that's very important to note is that I was promoted to professor a year ago, and Jeff has just heard that he's been promoted to professor, but his promotion hasn't yet taken effect. So he's still associate professor, Jeff Carroll. And if there's one take-home message from this session, it should be that. I think it takes effect in June, right, Jeffrey?
Do I need to call you Professor Wild for the duration of this talk?
At minimum, if not Professor Sir Lord Wild. Anyway, we do have some questions coming in. So thanks, guys. This is all Q&A. So if no one asks anything, or if we run out of questions, then the session ends immediately. We'll do everything we can to answer as well as possible. So the first question from Mary Dunleavy, and Mary herself admits that she was so excited that she hit send halfway through the sentence. The full question is, are there any trials where the goal is to reverse the HD symptoms? Which I think is a really useful starting point for a kind of overview of where we're up to in the world of HD therapeutics. So I guess all of the trials that are going on could reverse the symptoms, and the distinction is whether that's all they do, or whether they, which is good, or whether they are actually also aimed at reversing or slowing the underlying disease process. So we have this slightly artificial distinction between symptomatic therapies, which would make the symptoms less bad, and disease-modifying therapies, where the goal is to either slow the progression or even pause it or prevent the onset of the disease. So if we're talking about reversing symptoms, it's a slightly difficult question, because you know, something like tetrabenazine, which has been licensed for quite a while now, suppresses the chorea and could be thought of as reversing the symptoms, in the sense that you could have an amount of chorea that we call 10, that was before you take it, and then you take it, and then a week later you only have 5 chorea, and that would be a sort of reversal of the symptoms, and that might be helpful to the person, but we wouldn't have done anything to the underlying disease progression. So where should we start, team? Do we want to start with what's happening in the world of symptom treatments? Let's do that.
Sure.
So what's going on there? So symptom trials, these days there are fewer trials for drugs for symptoms. Tetrabenazine was licensed a few years ago, which is a modified form of tetrabenazine. Valbenazine is currently being tested, which is a drug that may do something similar to tetrabenazine. So that's another drug for the movement disorder aspect of HD. And as I understand it, that trial is expected to read out sometime next year. And I forget the name of the trial. Help me out if anyone can. Connect. I think it might be called Connect HD. That's a trial of valbenazine. That might be it. I can't think of any other trials that are purely about symptoms. Oh, there was a trial of bupropion for apathy, which I suspect may be fully recruited or have concluded, but I haven't heard any results from that. And then, of course, the other one that's been going on for quite a while is a trial of deep brain stimulation, electrodes into the head, putting electricity through the brain to try and help with the movement disorder aspect of HD. And I know that trial is still active and recruiting, and it's focused in Europe. But because it's such a invasive technique, the criteria are very tight, so that trial's recruiting slowly, which is not a surprise. Those are the symptom trials. In terms of trials,
if anything else, or if HDSA staff know any details, they can update us in the chat. And then we could talk a little bit about the trials focused on attempted disease modification, which is trying to directly impact the course of the disease. And I would say the most advanced of those are the various Huntington lowering trials. Rachel, do you want to explain the sort of background ideas and concepts of Huntington lowering, and then we can, as a group, kind of tick through the ongoing lowering trials?
Sure, absolutely. So I think most people who've tuned in today are probably aware that if someone has HD, it means that one of the 2 copies of the Huntington gene will have an expansion at the very beginning of it. And so this expansion is often referred to as your CAG number. And so anyone who has a CAG number over 35 will probably have Huntington's disease at some point in the course of their life. And so people who have HD will make two versions of the protein encoded by this gene. They'll have one, which is the normal protein that works nicely, and they'll make another version of the protein, which is this expanded form, which contains this mutation. And we have a lot of evidence to suggest that basically, if people are making this expanded or wonky form of the Huntington protein, it does lots of bad things in our cells, and it stops various biological processes that are really important from working. And then this is what we think is resulting in the symptoms that you see in HD patients. So one of the strategies that a lot of companies have think is very tractable for making a new medicine to treat Huntington's disease is to try and switch off or lower the amount of Huntington protein that is being made, and trying to, these are all kind of generally referred to as Huntington lowering therapies, but there's kind of a real diversity in the different approaches that these companies are taking. So some of them are specific, they only switch off or lower the amounts of the expanded or mutated form of the Huntington protein, whereas others will target both types of the Huntington protein. And some of them are reversible and others are, it's a one-shot therapy. I don't know, Jeff, if you want to take it from there, or I can carry on. Sure. And go for the different companies.
Yeah, I'll maybe try to tick through, and it's funny, but it's actually like it used to be just one or two, so it was easy, but we might even forget about one in this, yeah. Given that things are progressing really quickly. So I'll try to tick through, and then if people have follow-up questions about specific efforts, they can say in the chat. And I know that someone upstream had asked about kind of an update on the silencing trials. So the largest and most advanced trial for Huntington lowering, and this was a kind of drug called an antisense oligonucleotide, which is like a heavily modified stretch of DNA. That was pioneered by a company called Ionis, and went to the clinic with Ionis and their partner Roche Genentech. As folks may know, earlier this year, unfortunately, we got the bad news that the phase 3 study of that drug, which is a large scale study designed to see if it actually made people's HD better, not just if it changed this or that lab measure. That trial was halted for signs that it was not working, and that in fact on some measures may have actually exacerbated some symptoms in HD patients. So that trial was halted early, unfortunately. It's important to note that that doesn't mean that all antisense approaches, because there are other companies doing antisense approaches, it doesn't mean that all antisense approaches are a bad idea. So for example, Ionis, in partnership with another drug company, Biogen, just had a trial announcement from a genetic form of ALS, or Lou Gehrig's disease, and they didn't see any of the kinds of symptoms that we saw in this big HD trial. So I take comfort from that, in the sense that it doesn't mean that putting ASOs into people's brains who have brain diseases is a bad idea. It just means that particular drug at that particular dose didn't help HD patients. So still really terrible and difficult news to deal with that trial. But certainly I think not the last we'll hear of ASOs. And speaking of which, Wave Life Sciences is an ASO company that's currently running an ASO study in humans, and it's a modified version of an ASO, a slightly different chemistry, different target. And what's unique about Wave's approach is they're targeting just the mutant copy. So as Rachel explained, almost the vast majority of HD patients have a mutant HD gene and a regular HD gene. And the Wave approach targets some little coding sequences between the two. And that's cool because it just lowers the mutant Huntington ideally, and not the wild type. It's tricky because it means that you have to have the right other of these little coding variations that they're targeting. And so the consequence of that is not every HD patient is eligible for every one of Wave's style drugs. So that companies like Wave have the idea of having multiple of those drugs, and then hopefully each patient would be able to get one of them. So Wave is currently running one study with their new chemistry, their newest version of that ASO chemistry. Very excitingly, there are not one, but two companies that have announced the intention to run trials. They're not running a big HD study yet, either of these companies, but both Novartis and a company called PTC Therapeutics have announced really exciting approaches where a small molecule drug, which is a drug that you can be taken like a pill, like aspirin, that surprisingly and still mind-bogglingly, if that's a word to me, lowers Huntington levels. So it's an oral drug, you can take it as a pill, and it lowers Huntington levels across the body. Now this one lowers both wild type and mutant, but potentially could work as a Huntington lowering therapy as a pill. And so both Novartis and PTC are thinking about trials. And I'll pass it over to Ed because he's hopping up and down.
I just wanted to interject because it's very easy for us as the scientists who see this stuff kind of developing, and we knew it was coming. I think it's very easy for us to underestimate the magnitude of what you've just said. 5 years ago, because it was 6 years ago, because it was the only way that we could possibly contemplate doing it, it was the only, we could drill holes in people's heads and put in a gene therapy, or we could stick needles into people's backs and put in an ASO. And we did it because there was no prospect of a pill that could lower Huntington production, which is the thing we want to do. No viable prospect in the foreseeable future. 6 years later, we had some successes and we had some failures from those Huntington lowering programs. But here we are, like London buses, at the same time, 2 trials of an oral, a pill, or in one case, like a solution, a syrup that you swallow, and if they work as they have been suggested to in human beings already, that will lower the production of the protein that causes Huntington's disease. It's not a time where we get to hear a lot of good news, but this is a real kind of testament to how if we all just keep pulling in the same direction, the scientists, the families, the pharmaceutical industry, and everyone, and tries to just try and make the world a better place, eventually remarkable things happen. They might all fail, but my goodness me, 6 years ago, it was inconceivable that we would have 1, let alone 2, oral drugs to lower Huntington. And I'll hand it back to you to talk about those in more detail.
I think we can probably leave it at that. I think, these are closely related molecules that work via a mechanism that we think we understand at pretty high level of detail. So like Ed said, the devil's in the details, and you never know until you're in a large group of HD patients and you see if it works. I'm really excited. I'm very heartened by PTC made an announcement of their clinical trial design. And I think it's very patient focused and very, very smart in how they're designing these trials. I think the drug companies are working with HD patients. They're going earlier in the disease course. They're doing a delayed start design, so the people in the placebo group will eventually be put on drug, as has been done by Roche with the ASO study. But I think these companies are really designing responsive studies for HD trials. And I'll let Ed talk about the gene therapy trials, but I wanted to come back to an earlier question in the thread from Sophia Wood, who says, hi, I'm a math student, I'm at risk for HD. Are there ways mathematics or data science is used in research? So just to say that yes, and you can work on HD very usefully in that field. So lots of brilliant statisticians and people who work on clinical data have helped design the tools. So think about like how complicated HD patients are. You could measure any number of things, their apathy, their irritability, their movement, changes in their brain volume in very different ways. And there are these really sophisticated ways of kind of combining all of those data into a single score that we hope can tell us with much greater accuracy than just looking at movements or just looking at imaging or whatever, whether someone's getting better. And so PTC in particular is using one of these new HD scores derived by really smart statisticians to set what we call the inclusion criteria for who will be involved in the first of these HD trials with a small molecule lowering drug. So definitely, Sophia, plenty to do in the field and very usefully. And I've been talking too long. So either Rachel or Ed, if you guys want to talk about Unicure and gene therapy.
Yeah,
Rachel likes numbers, so you go.
Yeah, I was just going to add to your response to Sophia that one of the really emerging areas of study is not just doing the physical experiments, but it's also just this really, really large scale data analysis, like Jeff said, but using artificial intelligence and machine learning to really get a much better understanding of the details of all the data sets. And there's been so many advances in AI technology being applied to all different areas of medical biology. So if you have skills in maths and computer sciences, there's your skills are in very high demand right now as people try to tackle questions that we thought would be impossible because we just didn't have the computer skills or mathematicians to support those kind of projects. So in that respect, it's a really exciting time to be working in that kind of area. What were we going to talk about next?
Right, we're just going to kind of wrap up the clinical trial landscape discussion. And so I'll talk about Unicure, and then we'll talk about predopidine, because someone else has asked about that. But before that, just to kind of put a bow on the Roche discussion, the status of that program is that the treatment has been stopped in all arms of that study, but the trial has not terminated. So the people who are in that program are invited to keep going back to continue supplying samples and data to try and help figure out what happened and what happens when you come off a study drug and so on. So the other thing and that's an incredibly valuable thing. And so hats off to anyone who did it, anyone who was in those programs and felt they weren't able to carry on. Equally, hats off to everyone involved. This is the only way that we'll test whether drugs work. The important thing I think to remember from that program, which is a lesson across the board, is that a trial tests a particular regime that you put in the program, and it tests whether it worked in the people in the trial. And to get to the stage of starting a trial, you have to make careful decisions about dose, frequency, the participants, the age and CAG count of the participants, and so on. And once that's locked in, the trial tests whether it works in that group of people. And overall, the answer from the Roche program was clearly that the dose of 120 milligrams every 2 months or 4 months wasn't successful in slowing the progression of Huntington's disease in those 800 odd people that it was tested in. But the story is not finished for that drug. And how we know that is that at the HSG meeting a couple of weeks ago, or was it like 6 months ago? It's very difficult to tell these days. About a month ago, the HSG meeting, Roche gave an update, and the only real update they gave was that they aren't finished yet. And they're going to give a further update, public update in January. So, and that was the first time they've ever committed to a kind of timeline for the next big download of data. And what's been happening is because that program ended about 18 months earlier than it was supposed to, all of the samples have had to be shipped to the central repository and then shipped out to the labs that analyze them. And suddenly all of this data landed 18 months, and it takes such a long time to plow through that data and make sense of it. And the question is, well, we know the answer is no as to whether that drug worked in that trial. But the question is there a strong scientific biological reason to believe that there may be a group of patients in that program who did benefit from the drug? And if so, is that enough to take it into another trial? Or, could you modify the dose or the regime in some way? And I don't know the answer to that yet. No one does. If Roche already knew the answer to that, they would have had to announce it publicly already. So the fact that they've said, we're going to tell you in January, means they're going to tell us in January. So it's worth, bearing that in mind, that there's sometimes a phoenix rises from the ashes, or the cloud has a silver lining. Take your pick. On to the other stuff, though. So we now live in the age of gene therapy for HD, and it may sound like injecting DNA into someone's spinal fluid is gene therapy, but it's kind of not. It's not defined as a gene therapy, because the ASO trials inject DNA, but the DNA behaves more like a drug than an actual gene in those participants. And if you stop giving the injections, it kind of dissolves away. Gene therapy is where you take a friendly virus, scoop out its contents, replace it with new DNA that's an actual set of instructions, and then those instructions get into the brain cells via the virus, and then the brain cells start making new stuff. In the case of the Unicure program, what they're making is a gene silencing drug. So the virus infects the brain cells and is injected through an operation, a neurosurgical operation. And then it turns those cells that it infects into a factory for making a gene silencing drug. And it's making that drug every day, potentially for the rest of the person's life. So it's potentially a sort of high risk, high reward strategy. And the news, if you're doing that kind of stuff, major operation, and it could last for the rest of your life. Clearly, that's an approach that requires caution. So quite rightly, these are trials that are very, very, very slow to build up, slow to recruit, and they follow the patients up for a long, long time. And if I haven't lost count of the years, we're now 2 and a bit years into the age of gene therapy for Huntington's disease. That's two and a bit years. Is that right, guys? It's not 3, is it?
I don't think so. And did you
I
mention
think
it?
it's 2.
They just
And
got
basically-
permission to start dosing the
Yeah,
higher level
exactly.
in that trial?
Yeah, so we get intermittent updates, public updates, every time basically the safety committee has met and has given the organizers permission to either open up into a new cohort of patients or to increase the dose of therapeutic that's given. And in about October or November, Unicure announced that they had been allowed to start dosing at the higher dose. Someone's asked in the chat, someone who's clearly very in the know in the pharmaceutical world has asked, has pointed out that Unicure promised that they would share early imaging and biomarker data from that program before the end of the year. And have asked whether there's any prospect that that might tell us that the drug is that the program is working. I would say that's the kind of news where, well, so the nature of a trial program like this is, if there were significant safety concerns, they would pull the plug and we would have been told about it already. So the only question is, can we get a good signal from those very early data? And I would say, don't hold your breath, because we're talking about tiny numbers of people, and we're talking about drug that was given at deliberately, or not drug, gene therapy agent that was given at deliberately very low concentrations to gradually build up a safety understanding of the approach. And also, biomarkers like neurofilament and brain imaging, it's possible that those could be kind of altered in ways that are difficult to understand by the process of drilling holes in someone's head and infecting the brain cells with the virus. So what I would say is, we sort of know, the one thing I think that would be really good is if someone's neurofilament level, which is a protein that's released when brain cells are damaged, if that level goes down from baseline to follow-up, I think that means that you've rescued neurons, you've rescued brain cells. And if that does happen in the data release from the Unicure program, then I would certainly view that as extremely exciting. What I wouldn't say is that the flip side is true. It's not bad news if the neurofilament level hasn't gone down. It could just mean that the small numbers of people low dose, and we need to follow them for longer. So and I wouldn't read much into brain imaging at this point, just because it's such tiny numbers of people, and you do need much bigger numbers for imaging than you do for something like neurofilament. So that's my kind of steer on that. I'd be very interested to see the data, but I think potentially more important is the ongoing news that we get every few months that the program is proceeding to the next step, which is kind of how it needs to go with these gene therapy trials. And one final word I think on prodopatine would be the Proof HD study. And this kind of takes me back to the things that we were saying at the start, which is there are drugs for symptoms and drugs that slow disease. And prodopatine kind of spans those two worlds in a way, because it was developed as a treatment for the movement disorder in HD, so a symptom treatment. And along the way, a suggestion has emerged from the people who make it that it might actually be able to have an unexpected impact on the progression of the disease through a mechanism involved in how the cells actually function and stay alive. So the new trial, which is called Proof HD, I believe has just been fully recruited in the past couple of weeks. I think I saw a press release to that effect. So that's good. Any trial that's happening, it's better when it's fully recruited than when it isn't yet fully recruited. And we look forward to seeing whether that trial, which is designed to detect whether the drug can slow the disease as opposed to just making a difference to movement disorders. Look forward to hearing the results. But once it's it's kind of like slow cooking a stew or a side of beef. There's a lot of prep that goes into it, then you fill it up, and then you have to put the lid on and leave it for a long time. So the lid is on, and that trial is now cooking. We might not hear anything for a while.
Sandra Lefko asked about if there's been recruited starting for the PTC trial. As far as I know, that's not the case. I don't think that they're at that level yet. If I'm wrong about that, Sandra, we'll put a tweet and link to the announcement, because there will have been a press release if they started recruiting. But I'm pretty sure they're not quite there yet.
Was that PTC, Jeff? Sorry, I lost you for a sec.
Yeah.
No, PTC not recruiting yet. That trial protocol is in the stages of being finalized and sent for regulatory submissions to FDA, EMA, MHRA, and so on. So that's, but I would expect, I'd be surprised if it happens if the first patient's recruited this year, but I'd be very surprised if it's not recruited in the first few months of next year.
So Ed, there's a question up thread that is a little personal, so I'll obscure a little bit of the details, but the gist of the question is that they're an at-risk person with a CAG of a certain size, and they're debating about how and whether to have kids and what impact the clinical trials that we're talking about has on that decision. And obviously that's a very personal decision, but I just want to share some experience. So, folks don't know, I come from an HD family. My mom died from HD. I inherited the mutation, as did several of my siblings. So, I'm in a very similar boat, I imagine, to the person who asked that question. And I'll just say that, based on all of the science and all of the clinical research that we're talking about, I am extremely optimistic, and it is a different universe, inconceivable from where we were when I had my kids 15 years ago. Having said that, when I found out about my mom getting sick, I had multiple professionals tell me that we were, 3 to 5 years from effective treatments for HD. And so from my point of view, until one of these things works, it would absolutely be my strong suggestion that folks not consider all this good news as a reason to say, oh, well, I don't have to worry about, the future generation. I think we have to worry about the future generation until we have definitive evidence that one of these things is seriously changes the course of HD. And my wife and I were lucky enough to do a successful round of pre-implantation genetic diagnosis, which is a technique to screen embryos for HD before they're implanted and result in a pregnancy. And so basically allows you to have HD-free kids. And if I was personally in a different life position, I would do PGD today, even as excited as I am about all of these trials. So that's not to say I'm not excited, but it's just the reality is we're trying to do something no one's ever done before, which is cure a brain disease like this. And I think we shouldn't assume it's going to work until we have evidence that it works. So that's my personal advice, is that I would do PGD today, even as excited I am as about all the science.
Yeah, I mean, I would share Jeff's optimism that, this is a really exciting time and there's so many trials either ongoing or about to start, and we're in a very different landscape than where we were 5 years ago. But I think something else to also touch on that I think is giving us a lot of optimism is everything that's happening before we get to the clinical trials. And there are so many advances, not just in Huntington's disease research, but in general biomedical research and new ways that we can tackle genetic illnesses. And one question here is from Rick, who is reading Science Daily and read about CRISPR-Cas9 as a way to edit broken or mutated genes. And this is like a really cool technology that was really not discovered until very long ago. And scientists are still trying to figure out how we can use it to help treat certain diseases. So just a bit of background, this is very intricate molecular machinery, which is able to go into our genome and very precisely edit certain changes that you might want to make to that. So you could envision, for example, that you might want to go into the Huntington gene and reduce the number of CAG repeats that you have from a number that would mean that someone would have Huntington's disease to a number that would be within the normal range. And that's work that a lot of people are doing, and there's a huge amount of research being done right now on using that precise technology with that endpoint in mind. But it is a long way off, and we're hopeful because all these new tools are giving us new answers to how we might be able to tackle Huntington's. But CRISPR to treat a brain disease is probably not going to be available just yet, but there's a lot of hope for things like, for example, scientists using this to treat things like blood diseases, where you do essentially a very complicated form of dialysis, like taking someone's blood and then doing the edits on the blood, and it's outside of the body, and then returning the blood back to the body. And that's something that can be done quite conceivably. We just don't have the technology yet to do that for a brain related illness, but that doesn't mean it's not going to happen. And more recently this year, we wrote on HDBuzz about a different type of technology altogether, which is almost like we referred to it as a dimmer switch in the article, about a new different way that you can change the level at which you have a gene expressed. And so there are all sorts of people who are working well before the clinical stage of science to using all these new technologies and inventions and discoveries to see if they might be amenable to make new drugs and medicines for Huntington's disease. So, you know, it's good to know that there's all this exciting stuff going on, but we're not putting all our eggs in one basket, and people are trying lots and lots of different approaches to tackle this disease, which is, you know, probably the best way to go about it.
Cool. So slightly going back to what Jeff was talking about with, you know, planning ahead and trying to figure out what's on the horizon. You know, there's a related question about observational studies and who can participate in those if they haven't been tested for the HD mutation. And to me, this touches on the kind of the core fact that we all are on the same mission. And there isn't really such a thing as an HD scientist versus an HD family member, you know, or, you know, we're basically all team members in the same team, and we may have different roles at different times. But this is the mission, which is to make the world a place where people don't have to worry about Huntington's disease anymore, is one that will only be accomplished. I think it will be accomplished. It will only be accomplished if everyone who can help does help. And the more people who are helping at a particular time, the more quickly we'll reach that point. So I think we'll get there. The only question is how quickly. And so I guess the question sort of touches on what's the point of observational trials, which is, you know, studying the disease in humans. And that's things like Enroll HD, HD Clarity, Prevent HD is another one that was mentioned, and you know, names like Track HD and Cohort. Those are all observational studies because there's no experimental drug given. What's the point of those when we got so many therapeutic trials? Why don't we just keep doing more and more therapeutic trials? Well, I think the answer is that partly we are where we are with the therapeutic trials because so many people took part in observational trials in the past. So a very vivid example is that in the next year or so, we expect a trial of a medication that targets the apparatus that repairs our DNA. It seems that behaves differently in people whose HD starts earlier than expected or progresses more rapidly than expected. And so this trial is basically trying to adjust the behavior of the DNA repair equipment in a way that should hopefully prevent or delay the onset of Huntington's disease. And it's doing it at a really fundamental level in the HD gene itself. It's a really kind of innovative approach. And it only exists because 20,000 people gave blood for the Enroll HD study. The DNA was extracted from the blood. The DNA was then studied. And that DNA, the variations between people's genes, not the HD gene, but other genes that they happen to carry, those variations pointed us towards this pathway of DNA repair as a potentially very important thing in Huntington's disease. That's one example. Another is my work in biomarkers. So basically, you know, if someone with the HD mutation who doesn't have symptoms, if I give them a drug today, and then I check back in 5 years and they're the same as they were today, but 5 years older, how do I know if that's because I gave them the drug or were they going to look fine anyway? Because we know that people look fine for many years in many cases with HD. So that's why we need these biomarkers. And that's basically ways of sort of looking inside the brain without having to drill holes in the brain. Markers of brain health, in other words. So a neurofilament is one of these. It's something that we can measure. And if it goes up in the blood or in the spinal fluid, it usually means that the brain is unhappy, it's not doing well. If it goes down, it usually means that the brain is doing better than before. And we can only start to use those biomarkers to run trials in earlier and earlier in HD if we have enough data to support their use. And that's why we need to keep doing these trials like Enroll HD and HD Clarity. You know, there's obvious reasons why people are excited about taking part in a drug trial or trial of a new drug. You know, you might be given a drug that works. Even if it doesn't work for you, it could work for other members of your family or the future generations. And of course, you know, I think it's appropriate that therapeutic trials are everyone's top priority for getting them recruited. But, you know, not everyone has access to those trials. Many people who want to be in those trials are not eligible, either because the trials are full or because they're too young, too early, too old, too male, too female, whatever. Their cholesterol's too high. There can be all sorts of reasons that vary from one trial to another. I would urge everyone, as a matter of course, to please volunteer for every observational study that you're eligible for, with a focus on Enroll HD, because that's the biggest study there's ever been in HD. And it's the one that serves as a platform for other observational trials like HD Clarity and for drug trials as well. So it's a kind of recruitment database for future drug trials. The specific question that was asked by Kelly was whether observational studies are open to people who haven't been tested for the HD mutation. And some of them are, and some of them aren't. So Enroll HD is the big observational trial, and you certainly can be involved in Enroll HD if you haven't been tested. And how that works is that they take a blood sample, they replace your name with a code number in a lab on the other side of the world, and a test for the HD mutation is done. But that result is then never fed back to your clinical team that's looking after you. And even if someone broke into that lab in Germany, is it in Germany? Anyway, wherever it is. If someone broke into that lab and found the data, found the code number, they'd have no way of linking that number back to you in your home. So there's no risk of accidental disclosure. There's an infinitesimally small risk of accidental disclosure, and it hasn't happened yet in tens of thousands of participants. So that's, I would say, a safe way to take part in observational research. And also being in a study like Enroll HD will bring you into more, frequent contact with HD researchers. So it's a good way to kind of stay in touch with what's happening.
So Alyssa had a good question about sort of in a different area entirely, which is about which efforts are being made to connect with our HD families in underrepresented countries. From my reading, I'm not seeing much efforts for work, for example, in the continents of Asia or Africa, which is a great question. And it's something I assure you HD scientists have talked about, certainly at meetings I've been, this has been a conversation. And part of it is a broader conversation about expensive therapies like ASOs. It's very difficult to deliver these things, and there's a certain level of sort of clinical experience with these kinds of drugs that has to be there to actually run the trial. But it doesn't mean that we shouldn't be interested in the experience of HD families in these countries. And just to say that this is improving. So, for example, I was present at the first meeting of the first HD network in China. And one thing here is also a genetic issue, which is that it just so happens to be that as far as we've looked, which is not far enough, it's true that HD is more common on Northern European backgrounds. So it does seem that among, say, European populations and European-derived populations in North America, HD is more common than, for example, in Japan, where it's very rare. And as it is in China, just by virtue of the genetics of those populations. But China's huge, and so even if HD is much rarer there than it is in the US, there are still many HD patients in China, and they deserve just as much attention and care. And in fact, I was very heartened that HD clinical network was was brought on to be part of the Roche study. So there's, I would say, progress in this direction. Obviously not enough, and we're not done. But I think scientists are aware that we need to expand our look beyond white guys, said the white guy, and hopefully we're getting a little better. But it's a good question and something we need to get better at.
It's worth mentioning also, Jeff, that these things do take a while to gather momentum. But since I've been in HD, it's 16 years. But I think about 10 years ago, a new network for Latin America was set up, the Red Latino Americana de Huntington's disease, or RLAH. In the past 5 years, there's a new network in South Korea. I've been involved in helping Nikhil Ratna to set up a Huntington's disease network in India, where I think there's a lot of HD, much of it undiagnosed, and clearly it's a pretty big country. HD is probably as common in India and South Asia as it is in Europe and the US, probably. But that network is gathering momentum and is certainly active. And the final one was Africa. Could I forget? So in the past 12 months, I've started seeing tweets from an organization which is an HD network in Africa. If anyone has a specific, desire to be connected with any of those networks, you can send us a message through Twitter on HDBuzz or send one to me on Twitter, Prof. Ed Wild. It's happening. It does take a while.
I wanted to jump in and to go back to something that Edward mentioned about sort of observational studies and their importance, and just sort of highlight, we actually have an article coming out on HDBuzz, hopefully this week when I get my act together, about some really cool tools that people have developed to basically allow us to better understand progression of the disease. So you'll hear a lot about these different biomarkers, and a lot of them are taken from either spinal fluid or from blood, and then scientists are taking those samples and measuring the levels of different proteins in them. But all of those are really proxies for what we want to know is what's going on in the brain. And so more recently, some really cool scientists from labs across Europe and North America have made these very clever tools, which basically can be ingested by a patient, and they will show you exactly where certain clumps of Huntington protein will appear in the brain. And these clumps of Huntington protein in the brain are something that build up. We know they build up over time, but they're very, very difficult to measure until either you have to do it by looking at an animal model, and you have to wait till end of life to be able to see them so that you can actually image them, whereas this is a really safe and awesome procedure which allows you to see them in real time in a patient through a very simple brain scan. And so there's technologies that are not just being developed for therapies and cures, but also for us to help understand how the disease is moving forward. And you can imagine that these kind of tools could be used to help see if treatments are working. So we can look at these special tools which will light up these parts of the brain, and if the way the brain lights up changes over time as people are taking treatments, that could be a really helpful way for scientists to understand how well medicines might be working. There's another question here that I think is probably best for Jeff, which is about animal symptoms in Huntington lowering therapies, because there is, what are the indications about whether drugs are working in animals or HD models?
Yeah, so it is a great question. Thanks for highlighting that, Rachel. Again, from Sophia. So the question is about, the extent to which Huntington lowering, these various Huntington lowering drugs have been shown to completely reverse HD symptoms in a mouse model. And as someone who's spent 10 or 15 years or so working on HD mouse models, I'm now going to tell you how bad they are. So, the real problem, and this is not just HD, this is generally, mice are not humans, right? They're extremely useful. We can do lots of things in mice that we can't do in humans. We can access their brains. We can give them drugs that would be unethical for humans. There's a lot of reasons we use mice, but they're just a tool. So the key thing, if you think about HD in your mind, you visualize an HD patient that you love, the first thing you'll think about is the movement disorder, right? Most HD patients have chorea. They're twitchy, they're moving. Mice don't get chorea. There is no mouse model of HD that has chorea. So there's a very big disconnect between exactly what happens in the humans because of HD mutation and mice. And secondly, mice only live a couple of years. So we kind of have to crank up the HD mutation or express a lot of the extra of the HD gene to make them have symptoms. And we've gotten much better at this now, and we have better mouse models now than we ever did. But just to say, even as someone who thinks a lot about mouse models and values them, I think we should take the data that we get from them with a little bit of grain of salt. Having said that, I think there's been kind of a range of experiments that have been done. Unfortunately, there hasn't really been a systematic, careful head-to-head comparison of, say, ASOs versus gene therapy. In general, all of the Huntington lowering studies that have been published have shown pretty reasonable effects on the symptoms that look like HD in mice. I wouldn't say that it's a slam dunk, all the symptoms go away or completely reverse. But again, these things we're measuring in mice, changes in their brain chemistry, loss of the synapses in their brain and things. We don't know exactly how it paints onto human HD. And so we use mouse trials to help us plan our studies, but we shouldn't put all of our eggs in the mouse basket. That sounded weird. Did that make sense?
That's excellent.
Something about a mouse egg basket. Jeff, the simple core of the question, which you've circumlocuted around, was has a disease ever been stopped completely in a mouse or even reversed?
With genetic techniques that aren't what we're doing in humans, you can reverse the aggregates in the brain and some motor symptoms entirely. That's been known for 20 years. But that's a sort. I don't like to focus on that because a little bit of it's a complete shutdown of the expression of the mutant HD gene in basically every cell of the brain, which is sort of a kind of a perfection that's quite easy to do in mice, but virtually impossible to do in humans.
A couple of questions from Laurie Powell. The first is about, she asked a couple of specific questions about whether, about what medical conditions might prevent someone from being in a trial for HD. And it's an important point, but I'll answer it in general terms rather, because we're not here to give medical advice, but in general terms, the trials will be set up with strict inclusion criteria and strict exclusion criteria. For instance, someone who doesn't have the HD mutation can't be in a trial of an HD disease-modifying drug, a Huntington-lowering drug, typically. There are some softer criteria, and the exclusion criteria are often things like you can't be in a trial if whatever. So if you're in another trial at that moment, or if you've had a major spinal operation in the past 5 years or something like that. The more detail you go into, the more variability there is between different studies. And there are obviously also medications that can prevent people from being in trials. It's very difficult to be helpful in terms of giving generalities. What is typically the case is in a trial of Huntington's disease, if someone has another major brain disease, like a stroke or a brain tumor or Parkinson's disease as well as Huntington's disease, typically that means that person, the drug might behave differently in that person's brain, and therefore they're unlikely to be able to take part. Cancer and other big physical medical conditions, like I can't think of any, liver failure, heart failure, big medical conditions typically would mean that your body's going to be behaving differently, and that means that the data that you give, even if you behave immaculately during the trial, the data might not be so helpful when it comes to understanding Huntington's disease. So, but when you sort of start to come down the list, so for instance, well-controlled diabetes, maybe not, skin diseases, or the presence of a particular medication or another medication, the answer is basically you've got to speak to the trial team. The trial team may be able to give you an answer straight away, and if they can't, they can speak to the medical monitor for the trial. The bigger picture, though, is try to be a bit JFK about it. Ask not whether you can be in a particular trial, but rather ask, how can I help? What's the best way for me? This is you speaking to your HD team. What's the best way for me to help the overall research in Huntington's disease? Is it for me to be in Enroll HD or HD Clarity or a particular drug trial? Or if not this trial, then what other trials are on the way? But also think beyond yourself to how can I help other people be in trials? And Malaria, from your question, I can see you're already doing that. But that's my sort of general advice. Try to be, instead of asking about one particular trial, although it's important to be enthusiastic about individual things, that's kind of our whole thing. Try to think more generally about what's the most useful thing I can do. And you also asked, Laurie, about what are the main barriers to progress in Huntington's disease. I think money is always an issue. Huntington's disease, thanks to CHDI Foundation, is much better funded than other similarly rare diseases, but more money is always better. And what we don't have is enough sort of public money from big government funders coming into HD. And also, I think in some countries, the USA among them, the healthcare infrastructure is, to put it generously, I think, highly variable from one part of the US to another. And that makes it very difficult for people, because people access research participation through the HD clinics. The HDSA and others are amazing at putting in place the best infrastructure they can. But I think that's a limitation. I would also say that the number of people who participate in research is a tiny fraction of the number of people who would eventually benefit if the research develops drugs that would be helpful. In many cases, that's because the people involved aren't able to participate practically, and we need to do everything we can to reduce barriers to participation. But for instance, trials in pre-manifest HD haven't really been a thing until now, but with PTC, that trial is going to be the first disease-modifying therapeutic that's going to be tested really in largely people with pre-manifest or people who don't yet have clear signs and symptoms of HD. So I think it's about doing whatever you can to take part in as many people as possible, starting with things like Enroll HD, and take it from there.
Yeah, just to give some personal color to that kind of experience, I had a family member who's done an enormous amount of observational research for HD. They were in Track HD and Track On, which were very intensive, observational studies, contributed a lot to the HD field, wanted to be in a study with a drug, and found out in the screening process that he had this like benign brain thing that makes his MRI scans look funny. So it doesn't matter for his health, but it makes his MRI impossible to use in the study. And so they said, listen, we can't have you in the study because of this. And it's so frustrating as a person, as a family member, to hear like this thing that doesn't matter for you medically is going to keep you out of the study. But the reason is that those MRI results are going to be a key outcome of the study, and having someone in there whose MRI isn't interpretable, by the algorithm they're going to use is going to mean there's one less participants in it, which is going to make it a worse trial for everyone. So those kind of inclusion exclusion criteria can be really excruciating and really sad. And I know that personally, but it's for a good reason, and it's not because scientists don't care about those people, including our actual loved ones. So it's tricky, and I'm going to leave that comment. Unfortunately, folks, I have a classroom full of students really excited for this final exam I'm about to give them. So I need to leave at the hour here about now, but it sounds like other folks might be able to stay a bit longer. But I wanted to take a chance to say, I hope everybody has a great holidays, and thanks for coming.
Great. Bye, Jeffrey. Good luck. Be nice to your students. Rachel and I think can each stay on for about 10 minutes longer, if that's helpful. I want to ask Rachel, I'll put you on the spot here, but
okay,
if you don't know, you don't know. I think you may have written the HDBuzz article, but I can't quite remember.
Maybe.
Talk to me about thiamine and B vitamins and mice and people and Huntington's disease. There was a trial out of Spain, I think, that looked at this.
Yeah, so I think my understanding of this particular study is that there was a paper published, I think it was, like not that long ago, like maybe a month or two ago, in Science Translational Medicine, which is a very good journal, where they had looked at treating HD mice with thiamine and biotin. And they basically showed that in their mouse model of HD, that they saw some improvement of the symptoms of HD. And they think that this might be interesting to push forward into patients. And I believe there is a clinical trial ongoing in Spain, which is looking at treating patients with these supplements. Because, these are not drugs, these are supplements, essentially. And seeing
these are like vitamin type things, right,
exactly.
Rachel?
Yeah.
So
this is the thing to remember, right?
Sorry, go ahead.
I'm just going to say, so this is the thing to remember with stuff like this, is that often when we're designing drugs, when we're doing this kind of like ASOs, for example, or the splicing modulators that people like PTC are making, these are highly specific molecules which are targeting very specific mechanisms in human biology to give us the outcome that we hope will be, improvement of patient phenotypes and symptoms and so forth. And hopefully everyone will get better. But with things like vitamin supplements, vitamins obviously are very important in our general health, but these are very promiscuous molecules, and they're going around, they're doing 10,000 different jobs all over our bodies. And so it might seem that like sometimes giving these supplements is good, and it seems to be doing something great in a model, but whether that really translates to actually helping patients in real life is often very difficult to determine. And to the best of my knowledge, nearly all supplement clinical trials generally actually have worse outcomes for patients who take them, because you're giving such high doses of vitamins that it's generally not always the best. Although the side effects are generally pretty mild and not too bad. But I would be very cautious of these kind of studies, because most of it is done in mice. And as Jeff said earlier, mice are not people. Their brains are incredibly different to our brains, thankfully. And so we just have to be a bit cautious when looking at studies like this. It would be amazing if we could cure all diseases with vitamins, but there's a reason that hasn't happened so far. Vitamins are very good for us and they're very important, but it's unlikely that they will be the answer for something as complicated as Huntington's disease.
Yeah, it's one of the weird things about the mice, isn't it? That they're super sick. Like the mice are sicker than most HD patients if you scale it up to human size. But yet they seem to be easier to cure or easier to treat. Like we've had lots of examples of something that works really well in a mouse. Unfortunately, as we know, 100% of those has not worked when it was tested in humans. And I think that's a really important thing to bear in mind. Like we think that treating mice is a kind of necessary step along the way, but so far we've yet to actually go from that to success in people. I think the other thing about this biotin thiamine thing is that these are kind of vitamins to do with they're sort of B vitamins, so they're to do with the way the cells use energy and stuff. It's not the case that the study found that people with Huntington's disease are deficient in vitamins. If you happen to have a blood test or some other test and discover that you are deficient in a vitamin, that's when a vitamin supplement's really helpful. Or if you have reason to believe, like, I'm a white guy living in Britain and look what it looks like out. Even if the sun was up, it would be grey. So I don't get much ultraviolet. And therefore, I take vitamin D in the winter to try and help my immune system work better to fight off COVID. I don't know if I'm deficient, but I have reason to believe that I might be. In that situation, taking supplements is a good idea. And the reason we do trials is, and this is a really important lesson from the Roche program for me, and we say all the time, we do these trials to test whether a drug is helpful or neutral or even harmful. And as you've said, sometimes it turns out to be harmful. And that has been the case with a couple of these supplement programs in HD and other neurodegenerative diseases. Just because something sounds harmless and potentially helpful doesn't mean that it will automatically not be harmful if you test it in people. So in other words, something that seems helpful can turn out to be harmful. So I would say it's a really interesting scientific finding that there seems to be a way that the HD mice cells were handling these vitamins differently. But it's really important to go through the process of testing that hypothesis in a well-designed clinical trial, like the one that they're planning in Spain. But we're not yet in a position to make a recommendation either way, other than the general recommendation that you shouldn't do things that haven't been tested. And the ways to look after your brain health in HD are the really, really boring ones, which is exercise regularly, have a healthy diet, probably don't smoke, don't be too into kickboxing, don't get your head kicked in every week, etc. There you go. That's my view anyway. Jeff, who's left the meeting, agrees with me, I think.
Yeah, no, definitely. I think the thing that is important with these kind of studies is that they can really highlight interesting areas of biology. So we know, like, for example, if this shows that biotin is important, where it's like, well, okay, well, how, why? Let's work out the molecular details of that. And then maybe we'll be able to design very specific drugs which will work really well to target that particular aspect of HD biology. So that's the kind of interesting and exciting thing to take away from these kind of studies.
We had a couple of questions about telemedicine and remote medicine, which is a really interesting one. It's one of the very few upsides to the COVID pandemic that doctors in general and neurologists in particular were forced to actually, familiarize themselves with technology a bit. I'm obviously a bit of a geek, but many of my colleagues are very old-fashioned. And for the first time, we've actually started delivering the majority of our clinical services over video consultation. And obviously face-to-face encounters are still very important, but I think we're probably, once COVID is done and whatever that means, we'll probably end up doing much more of a balance between face-to-face and in-person. How does this filter through to clinical trials is a really important question. Enroll HD and the observational studies. The issue I think with Enroll HD, I don't think Enroll is yet equipped for remote encounters, or it varies between sites. So some sites are able to do some of the assessments, like maybe some of the interview-based questions and the cognitive tests. But for instance, we can't do the UHDRS motor examination because that relies on sort of physically feeling how stiff a person's arms are. And also those trials, those scales were only kind of validated in the face-to-face context. And in order to have them be valid and useful, you'd have to then kind of study whether a video consultation is as good as a face-to-face consultation. So there's a lot of work that needs to go into it. The clinical trials, because those are very much more kind of fast-paced and time limited. So once you're in a trial for 2 years, whatever happens in that 2 years is the outcome. Those have tended to move a little bit more quickly and have tended to be a bit more flexible in terms of allowing some of the assessments to take place by video or by telephone. For safety purposes. We are certainly heading into an age where remote digital monitoring is going to be a big thing. And so some of you, particularly if you're in the Roche program, may have been asked to wear a smartwatch or carry a smartphone, or even there are some programs trying to develop, an app that monitors your HD symptoms on your own phone. Those are sort of called ecological measures. What that means is that they're kind of studying HD people inside and outside of trials in their natural home environment, doing the things that they normally do, going to work, sleeping. I needn't list them all. But those kind of ecological studies will, I think, in the next few years turn out to be really important because they're capturing the aspects of the disease that are difficult to access otherwise, and they're much more natural, realistic measure of people's day-to-day symptoms. And the regulatory agencies really like that kind of concept, that you're, instead of having a motor examination once in 2016 and once in 2018, and you subtract the difference and that's whether the drug worked or not, your motor symptoms are measured every day in different scenarios. And so I think it's a really interesting time to be involved. And so telemedicine and remote monitoring, I think you can expect to see a lot more of that. Eric has appeared.
I'm sorry that the way that we wrap this up is all of a sudden Kelly and I emerge from behind our black boxes on the screen.
There are much worse ways for bad news to be delivered.
But I did just want to jump in and say, first, thank you to everyone who was able to join us today for your questions, for your participation, and for your patience, because I know that these sessions are so great for all of us in the HD community, but unfortunately they are limited and we're not able to get to every single question that was submitted. So we apologize if we didn't address yours directly. That said, stay tuned because we will have future sessions like this, and we'll have the team and you can ask again. And for anyone who's saying, wow, that was so much information, I don't know that I really was able to absorb it all, we will be sharing the recording of this session, so you can always come back and check it out again. Ed, Rachel, and to Jeff, who had to go drop a final on a bunch of students' desks, want to thank you so much for your time today and for being a part of this and for everything that you all do for the HD community. Really, we appreciate it greatly, and thanks for being a part of this today.
Huge pleasure. We'll be back as soon as we can. So if you didn't get your question answered this time, ask it at the start next time, and we'll do everything we can to get through all of your questions. But some really great questions today. Thanks, everyone.
Yeah, thank you so much for having us. It's been a pleasure.
Happy holidays, everyone. Bye-bye.
Happy holidays.
Take care, everyone. Thank you.
Thanks, everyone.